Yes, in the previous, hello everyone. In the previous session, we discussed about the basic data structure, variables, and the NumPy array in Python. In today's session, we start with the pandas in Python. So basically, pandas is a very powerful library for the data analysis and the data manipulation. And pandas also provide easy to use data structures, as well as we quickly perform the multiple operations on these data structures. Now the data scientists use pandas for the different purpose. So basically pandas provide easily handle the missing data, and it uses series for one dimensional data structure and the data frames for the multi-dimensional data structure. And it also provides an efficient way to slice the data or for the selection and the indexing the data. And it also provide a flexible way to merge and concatenate or reset the data. So which type of data structure pandas provide? That are the series and the data frame. Series are basically used for the one dimensional labeled array, and it basically contains different types of data type, such as integer, string, Python objects, floats, etc. And the data frames are the two-dimensional data structures that are basically holds data like two-dimensional array or the tables with the rows and the column. Now, if we want to work with the pandas in the Python, then firstly we have to we have to install the pandas library in Python, because pandas and NumPy are not the inbuilt Python library. These are the external libraries which are used for the data manipulation and the data analysis. So for installing, we have to use pip install pandas. After installing the pandas, we have to import the pandas in our Jupyter notebook, or maybe it is the Python cell, or maybe it is the other IDs like Visual Studio. But just we have to import the pandas to work with the data frames. Now if we want to create a own series, so we use pd, because pd is aliases for the pandas library. Then we use pd dot series, and we have to put some number to series. We can use list, we can use string, we can use dictionaries and other data types for the creating the series. Here I am using integer. Yes.
मेडिकल टेस्ट इनसे करना इनके पास
Okay.
चाहिए ना कंप्लीट वाटर टेस्ट
Do you asking something from me? Are you asking something from me? Okay. Okay, now we want to create the series in the pandas. So we just have to write pd dot series and pass the numericals. So if when we call, the one dimensional array is created. So at the 0th index, we have 1.0 value. At the first index, we have 3.0. Now if we want to create a data frame means we want to create a two-dimensional array, or we want to work with the two-dimensional data, maybe it is table. Now I am creating a variable, that is the date, and the pd dot date range, and it is further we use for the indexing. By default, indexing is start from the 0 1 2 3. If we want to use own indexing, then we can use the pandas indexing step. So here I am using the date range as a indexing. So here I am writing date range method. Then I am putting some value. 2013 denotes to the year, 01 denotes to the date, and 01 month, and 01 dates date. Then period is equal to 6. Period 6 means कि you want to create 6 indexing, 0 1 2 3 4 5. तो अब at 0th index, corresponding index 1, second and third. So period is 6. You want to create 6 indexing. Now if your date index is created, now you want to create the data frame. So we have to use pd dot data frame. So data frame function is used to create the data frame. Then np dot random dot randn. So np dot random means कि we want to create from the numpy array. So that is numpy array with numpy array 6 comma 4. 6 denotes to the number of rows and 4 denotes to the number of column. So it basically provide कि we want to create some number that is the 6 comma 4. And indexing, we want to create the index with the dates, and the columns is equal to list a b c d. So then we want कि column name is a b c and d. So we put the a b c d, and we can also put the another name. So now after creating the data frame, we just call df. So then we call the df, this data frame is created. This is the first index, second index, third index, fourth, fifth, and the sixth, and their corresponding columns just we have created. Now if you want to create a data frame by passing the dictionaries of the object, where keys are the column name and the values are the columns value. Now pd dot data frame, then we just write the as the dictionary format. In the last session we discussed that dictionary basically contains key and their corresponding value, यानी कि key and their mapping value. So we take a as a first column, that is key, and 1.0 is their corresponding value. Same we create the second column, their name is b, and we want the second column as a date timestamp. We want data type as a date. Now the third, we create pd dot series. We want as a list of the numbers which are start from the range 4, that is 0 1 2 3 4, and the 1 denotes as the whole number are in the column are 1. And again in the d we want this is the multiple of 4, that is the third index 1 and or the 3. So 1 into 4, that means ki multiple. Or then we want d type is equal to int 32. We want in this column the type of the data type is int 32. Then e is equal to pd dot categorical. Categorical means ki there is no 0 and 1 value, that there is a some alphabetical values. That means ki we have the test train. We have the string type value in your data, and f is equal to true. So basically from the dictionaries we create the data frame. These are these steps ki how we can create our own data frame by help of pandas. Now if you want to check the data type of the data frame, you can use df, the name of the data frame, dot d type. It basically give you the data type in your column. So a is the float 64, that a is the float 64 means ki it is a floating number. B is a date time 64, that means ki b is the date time data type. So this basically give you the type of the column. Now the second step is कि if you want to view your data, कि if you have the data and you want to view, so you have different way to view the data. कि if you use df.head, so head basically give you the top starting 5 rows, and if you use df.tail, so df.tail basically give you the last 5 records of your table or the data. Now if you want to fetch this something but starting, so you can put the value ki basically if I run this cell, Okay. Now df dot head give you the top 5. If I put one argument, we want only top 3. So basically it give you the top 3 records. Sometimes we have the large amount of data and we don't want to see everything in your data, but we want in the starting of your data, so you can use df dot head, that is the name of the data frame, and dot head and pass the value. If you want to fetch or you want to view the last records or last data, so you can use tail. Or if you put the any value inside the tail, so it basically give you the last 2 records. Again, if you want to display the index, if you want to check which type of index in is present in your data, so you have df dot index, which is basically used to view the index in your data frame. Now if you want to see the how many number of columns and what are the name of the column you want to see, and you can use df dot column. So df dot column basically give you the name of the column. Now one function is describe. So describe is basically used for the stat summary of your data. If you want to apply in the df, it basically give you the stat summary. Count means key in the a column you have 6 record, in the b column again you have the 6 record, but mean means it basically give you the mean of the a column and mean of the b column and mean of the data of the c column. And std basically give you the standard deviation. So in describe basically used to see the stat summary of your data, then maybe it is count, mean, standard deviation, or minimum, maximum 25 or 50% and 75%, to basically give you the stat summary of your data. Now if you want to transpose your data, suppose if your data is has the gene id and their corresponding samples, so genes are the rows and samples are the column, but sometimes you need you want samples as a row and the genes are as the column, so you read a csv file pd.read csv, then you see that in your data you have the genes as the row and the samples as the column, but you want key samples as the row and genes as the column, so you can transpose your data. So basically df.t is used to transpose the data. So and if you want to sort the data, sometimes you need you want to sorting, then you can use sort. The sort basically apply on the 2 ways. First one is on the basis of indexing. Second way is the on the basis of the values. So if you apply sort indexing, so
पर जब
axis
वो
1,
मानते हैं उसमें क्या आया था तो मैं
so
डालती थी
axis axis 1 basically denotes that you want to sort on the basis of the column. If you put here 0, that means axis 0 denotes that you want to sort the data in the basis on the basis of the row. So it basically 0, that is the if you doesn't mention any axis, by default it is sort the index on basis of row. Yeah, or otherwise you can mention 0. If you mention as a 1, that means you want to sort the data on the basis of the column. Then ascending means you want to if it is true, that means you want on the up, going up, but I am putting here false, that means it reverse the column, that means d c b a. You can also change ascending, descending, or the other way you can apply, you can play with the data. Now the this one is the on the basis of index. Now you can sort your data on the basis of the values. Values means you can put a number, like I put a number that is the by b. So by b means you want to sort the data on the basis of the b column. That means ki on the values of the B column. So in the B, we have the 0.3453, then 1.0. Now we have the number is minus 1.73 that are in the increasing order. That means ki B is short by the value. So basically you can sort the data on the basis of two-way indexing and the values. Now you want a selection कि you have a large of data, but you don't want to work with that complete data. You want some selection. You want to do something like you want only 5 features, you want only 10 features. So you just apply the selections for your purpose कि which type of data you want. So selections for the selections you can use get items. So get items are basically used to passing a single label. Single label means कि you just pass the column name. So A is just a column name, just a label of the column. So you can get like I want DFA, I want the data of A, then you can use the label A. Now if you also want the A, you can have the other method to fetch single data you by using dot method. So df dot A basically give you the same result as the df uh, double quotes A. Now, if you want to fetch the data but multiple columns, so you just pass like I want to fetch the data df, the class label, the column name B and the column name A. So we want two, you want multiple data. So you just pass the name of the column multiple B, A, C. Okay, so in this case, I am passing B and A. It basically give you the B and the A column data. Now, if you want to apply slicing, like in the last session we discussed about the slicing, you want subpart, so you just apply the slicing. So on this slicing means ki you want subpart ki start range and the end range. So in the DF, 0 means ki index 0, 3 means ki last index is 3. So 0, 1, 2. So first 3 is viewed as a result, or 3 means up to 3, not included 3. Now you want to also provide the range, like I would put the date time range to this range to this range. So if we apply this range to this range means ki you have applied the indexing is this range to this, so basically give you the result according to this slicing selection. Now the second way of selection is by label by using the LOC. So LOC basically used for selecting the row which matching the labels. So date 0 means ki if the index 0 is true, that it give you the details. So A, B, C, D give you the details of the LOC positions at index 0. Now, if you want to fetch the data of all but with the selected column labels, so you can also use df.loc, then all data, yani all index, but number of column is only 2. So that column indicate that you want all rows but with these corresponding columns A or B. So that are basically these are the selection area which basically choose on your purpose, on your problem statement ki which type of data you want to use. Now LOC basically use on the labels, that is the column name. Now you can select by the position. So we use ILOC for the position. So we put the value that is the 3. It basically give you at the index of 3. Now if these are the different way of the selection. Now you can also put the Boolean indexing. Boolean indexing means कि something is true or the false. कि if we apply 0 or 1, कि if we apply in the Boolean we have the true or false or the 0 and the 1 format. So I am putting here df, that df column A, the column A which in which the value is greater than 0. If these are true, that give you the answer. So we have the 6 row, but in the 6 row only 4 rows have satisfies the condition which is greater than 0. So it basically give you the range which is greater 0. And if we again also apply, so this, what is the basic difference between this df greater 0 and this df a greater 0? It basically apply on the select basic single row, basically on the rows type and individual values. But when we apply this one, this basically selecting the values from the whole data where the boolean condition is meet. So we apply a, so it basically check only for the a column. But when we apply the df greater than 0, it basically apply on the whole data set or all the columns. So we have the a b c d column, so it basically check for the all the columns where the condition meet, it give you value, otherwise it give you none for the which not satisfy the condition. Now again we have the another function, that is in method for filtering. If I'm just putting the df is equal to df dot copy to create a new data frame, that where I am attaching e. E is basically I am adding a new column in the data frame. So if df2 and I'm writing e, that means you want to add, this is one method for adding the external column in the data frame or your data set. So we use e, that is 1 1 2 3 4 3. So basically we put the values for the newly added column. Now in the df we have the 5 column in the e, which is the 1 1 2 3 4 3. Now I want to check which data is present or not. So is present, so is it means it is present, that it basically give you, if it is present, that give you the present data, otherwise all the data are discarded. So in this case I am putting the df, the column e, in the column e is in 2 and 4. If 2 or 4 is present, then basically give you the corresponding result of 2 and 4, and rest are the discarded from your result. Now you have the another thing, that is the settings. Settings basically used to new column automatically align the data by the indexing. If you want to add a new data and new column, then means that new column automatically align with the existing indexing in your data set or in your data frame. I am creating a S1, that is a series, and I want to put this series as a column of the data frame df. So df, f is the column name, and we put is equal to s1. S1 is the data. Now this one is the last column, e is the f is the last column which is newly added in the df. Now setting value, we can set the value by the labels. We want to change the value, we want to replace the value in the data set or the data frame. So df dot set means we want to basically mention the position. So at date, that is the index 0, a column is equal to 0. So at the first in index 0, a is 0. So if we call df, it basically gives the 0 0 0. Now again we can also set the value by position. So if we want to set the value by position, then it give you df dot iat. Iat denotes that if we want to put at the position. So at the 0th index, that means the row index 0, column index 1, that means column index 0 1 means 0 means a, 1 means b. Now we want to put at the row column is 0 and the column index 1 is equal to 1. Now the at the 0 index, that is the this one, and the 1 index of column this one, and we set or we replace the value by 1. Now sometimes we have the data but we don't know ki in the data we have the missing value or not. So to check the missing value, what we can do ki I just create a another data frame in which we have the missing value. For this, just simply add a column e is equal to 1 and rest are the if not satisfy the condition it give none. So now we want to check कि in our data set if any none value or the missing value present, so we can use the df1 dot drop na or how is equal to any. That means कि if any missing value is present in your data set, you just drop it, or you can call it delete from the data. But sometimes कि one we can handle the missing data. So we can handle the missing data. For that we doesn't need to drop the record from the data set. We just want to check if the missing value is present or not. So sometimes we want to fill the missing value with some numeric data. So I'm just simply taking here a value 5, but you can also apply how to fill the missing value. In the machine learning algorithms we have different, in the data processing techniques we have different type of methods to fill the missing value. For here I'm just putting 5. So to fill the missing value, df1 dot fill na means कि if any missing value present in your data, just put the 5. So in our data set at this position f we have the none value, we have the missing value, then that is replaced by 5.0, and in this case we have the null, then we have put the value 5.0. So just we want to put कि if we have any missing value then fill with this value. Now we want to check कि if in our data only want to check if missing value is present or not. So we can check for true or false. So pd dot is na and we pass the data frame as a argument df1, and it basically provide if the missing value is present or not. False means कि that is missing value is not present. At this position we have true, that denotes that कि here any missing value is present. Now we can apply several operations on the data frame. Just we can say that we can apply many stat operations on the data set. Now if you want to calculate the mean of each column, so basically you can use df dot mean. So it basically apply on the column, each column of the mean. And it means कि if a has this mean and b has this mean, so basically apply on the each column of the data frame. If you want to apply calculate the mean for each row, so you can use df dot mean and axis is equal to 1. That basically for the you want to calculate the mean value for each row. Now in the data frame you have the some user defined functions, that is the data frame dot aggregate, data frame dot transform. So data frame dot aggregate is basically used to aggregate the data. That means key to combine in one, apply on the multiple things, but combined result is given. So that means key you apply the aggregate np dot mean. So you want to apply on the a and want to apply the mean into 5.6. You also all multiply with 5.6. That means ki it basically agree. In the A column, every data is combined. Find the mean and multiply as the 5.6. That means it basically provide on the provide the result as the aggregated form. And the transform is basically applied on the whole data. If we mean, that means you want to apply on the this one individually. So basically it apply on the each value, that it basically transform the data. You don't want to aggregate, you want to transform. So it aggregate. So it means aggregate means ki you want to mean into 5.5, that means collective results, and you want to apply x into 101.2, that means ki you want to multiply that specific value, that means ki this value with this number. So basically it you can transform your data. Now the second term is merge. This merge is basically used to merging two things. Like we have the two data set and we want to merge into a single one. So you can use the different, you have two way, that is the concatenate, and the second way is the joins. Ki basically relational algebra joins. So you want to concatenate. So concatenate means कि you want to combine together the series and the data frame objects with the various kind of set logics, or you can simply say that the rows for column. So we create a data frame, then break into pieces कि we बस just apply the slicing df3 to 7 df7, that means all rows but 3 index, uh, only 3 rows, that is the start from the 3 but the all the columns. So that basically give you the answer as this df colon 3 df3 colon 7 df7 colon. Now you want to concatenate, then pd dot use the function concat and the apply the argument, that is the pieces, and it combine the whole data in a once. Now the joins. So how to apply the joins? The joins basically use to merge two tables, two data frames. So basically you use one data frame in which we have these data, second one we create these data. Now left in which we have this data, right table we have the data frame or the table we have this data. Now we want to merge left first table, then second, or on is equal to, we have to mention a column name or a key on which you want to join. So on is equal to key, that basically give you the this result. If you want to apply, this is the same result because we have the foo in the key, but if you want to apply on the unique, that means key we have foo bar 1 comma 2, foo bar r 4 comma 5. So if we want to apply the merge pd dot merge left comma right is on key. So we merge this table, that is the foo and the bar, and on the first table we have 1 2, on the second table we have the 4 5. So it basically give you the unique results. Now how to plot the results or the data? We use the matplotlib. So in the Python, we have two libraries, most popular libraries for plotting. First one is the matplotlib. Second one is the seaborn. So how to use matplotlib? The matplotlib is just using for using the matplotlib, we just install the matplotlib by using the pip install matplotlib. After installing the matplotlib, you just import matplotlib dot pyplot as plt. It is the default syntax for the matplotlib, and plt dot close all, and this one is basically used to close the window, the plotting figure window. Now I'm simply taking a simple example. We have a series of this number and now apply the cumulative sum and we want to plot. So this basically give you the plot of this result. This ts is the variable name dot plot, and this give you the plot of the data. If I'm not using this colon, there is difference. If I use the colon, that means this one is start from minus, then and increasing order. If I don't use the colon, it basically change the difference 0 minus 10 20 30. So which type of float you want, so you can apply on this way. Now you plot all the columns of the data set. You want to plot the comparison of your data, then you can use key if plt dot apply on the sum same on the cumulative sum, plt dot figure and df dot plot. And now we want to add as a legend. Legend means ki loc is equal to best. That means ki you want to apply on the all the columns. So legend on the a b c d, that is for that one. And same way if I am putting without colon, that basically give you the change the tick of the plot. Now this is all about ki we want to create your own data frame, you want to create on series. But if you have already the csv file and you want to import the data and you want to export the data, I'm just simply save a file with this data foo.csv. So we want to save the file csv. Now I want to read this file, so we have the syntax pd dot read csv. The pd underscore csv basically give used for reading the csv file. If you have the excel file, then you have to change the syntax pd dot read underscore excel to read the excel file. So you give with foo dot csv have this data. Now if you want to import the, you want to read the excel file, so you have the read excel, or you want to see the excel file as well. Now I'm showing you how data manipulation is applied and on a by using a use case. Here I'm taking a problem statement of breast cancer in which we apply the data manipulation and the data visualization. So how we apply this, we can see. Step first, so doing any machine learning task, firstly we have to clear that ki which type of problem statement we want to solve. Ki it is in the machine learning, if I think you know, in the machine learning, learning is of two type. Ki it is the supervised type or it is the unsupervised type. So for this, if supervised type, you have to go for the supervised learning, and or if you have the unsupervised data, you go for the unsupervised learning techniques. So that means ki in the supervised you want to do regression or you want to do classification. So just for doing anything, firstly you have to clear that which type of problem you want to solve. So in the specific use case, I want apply only data manipulation, but I want to further data classification. So for this, I am importing the important libraries. You all know pandas for the manipulation with using data frames, NumPy for numerical or stat analysis, Matplotlib, I'm using Matplotlib for data visualization, and Seaborn for the stat data visualization. Now, I am sharing some databases. If you want to practice ki which where I can download the data, so I am providing some links from where you can download the data according to your problem statement. So Kaggle is also a very large data repository on which different type of data sets are available. Next one is the UCI machine learning repository. It is the one of the oldest source on the web to get the data set. Now again we have the another repository, that is the GitHub. Or in the GitHub you can also download the different type of data. You can also download data from the Indian government sites and the US government sites and the British government sites and the French. But one thing to be noted, ki if you must just check for the data is publicly available or it is privated, for on the basis you can download the data. When we talk about the medical data repositories, so in the medical data repository we have different type of repository, that is the TCIA and the TCG, that are the most popular libraries on which we can download the medical data in the form of the genomic data, medical data, or the other type of data, like RNA-seq or the other type mutation type data. So we can download from these sites. And if you another site is the medical information mart for the intensive care, and the NCBI or NIIH data sharing repository. So for the machine learning purpose, if first thing is to collect the data, so you can download from these different repository for your purpose. Now the second thing is कि sklearn.dataset, कि if you want to work with the machine learning libraries, sklearn is the most important library to work with the machine learning algorithms, specifically machine learning algorithms. So you have used this sklearn. In the sklearn you have different type of things available. In the sklearn you have some default data set, you have also the stat related machine learning confusion matrix, you want to print classification matrix, you want to apply the data splitting, test, train. So all the things are available in the sklearn. And you want to load the models like logistic regression, random forest, all the models are present in the sklearn. At this step, I'm just loading the breast cancer data from the sklearn, that is the default data set. So from sklearn dot dataset, import load breast cancer, so that give you the breast cancer data set. Now I am importing load breast cancer. Now I'm assigning this data into a variable, that is the cancer is equal to load breast cancer function, from which is basically import from this library. Okay, after collecting the data, we know the data is noisy, we know data has some missing value. So data, we want data pre-processing. For the data pre-processing, we have the different type of techniques for the data pre-processing. First one is the data cleaning. For the data cleaning, we want to use if any missing data is present in our data set, so you can just ignore the you can remove that data, and simply you can fill the missing value manually or by mean or by most probable value. Mean means you find the mean of that column and apply on the none or missing area, and probably most probable value means ki that value which are appear in the most of the cases, you can pick that value and put into the missing value. Noisy data, so for the noisy you use these type like regression, clustering, or binning methods. Data transformation, ki if you want to apply transformation, ki you on the whole data you want some not specific on the whole data, some transformation, you want to change the reshape of the data, you want to change the data. For this you can apply normalization, you can apply attribute selection, you can apply these type of algorithms. Now you have the data reduction. So you all know that in the medical data you have the large amount of data, that means ki number of columns are too large. So suppose I have a data set, RNA-seq data set, and I have a 10,000 20,000 genes in the data, but I don't want to work with 10,000 or the 20,000, but this high dimensional data just basically high the cost of the computational cost of the computation cost of my algorithm or my the model, but just increase the, but I want to reduce the dimensity. So for reducing the dimensity we have different type of techniques. We have the data cube aggregation, attribute subset selection, numerosity reduction, and dimensity reduction. Dimensity reduction is basically most popular in the machine learning, unsupervised learning, PCA, TCNA, or the in the deep learning. If we want dimensity reduction in the deep learning algorithm, the autoencoder are used for reduce the dimensity. So for the exploratory data analysis, first thing analysis ki the data is univariant or the multivariant, or for feature engineering means ki which features is most appropriate for our problem statements. So just check that features. So we apply the feature engineering. In our data set, if we have the outliers, so just detect for the outliers and remove the outliers. If it is not appropriate for your data, so you can remove the outliers. Now in the cancer data, just we have load, we in the cancer data we have these things. Cancer, that the target. Cancer means data means ki you in the data we have the corresponding features data, and the target means ki you have two target, that is the malignant and second one is the benign. So you want to classify the in the data ki that breast cancer is the malignant or the benign. Okay, now I want to check for the keys, which type of data is present. So in the this file keys, we have data, we have target, we have frame, we have target name, we have the description of the data, we have the feature name, file name, and the data module. Now if you want to check the description, so basically this is nothing, we already discussed in our section ki how to call the data frame. So this cancer is a type look like as a data frame, so we want check for a specific column. We want, so that means ki you want to print cancer, that is data frame name, description is the column name. So we just want the selection of on the basis of class label, column label. So how to apply pandas and numpy in your problem statement? So in this way we can apply the pandas in our problem statement. Again, if we want to apply the print target names, ki which type of target, which type of class label we have. Target name means ki you want to classify, so from which it is a binary classification. We have two class, we have target label is malignant and the benign. So benign and malignant is basically two type of classification. We can say that is a binary classification. Now we have the cancer and then the data frame name, then the column name is target. Target means ki you have 0 1, so this is nothing but it is the binary format or the categoric format of this one, ki we want to label into the form of 0 and 1. So that means ki we have the target name malignant and we have the benign. That means ki we want to we have target is 0 1. 0 means malignant and 1 means benign. So we have the target for in this data set. And the feature names, ki this is the feature column in our data frame. So which type of features? So features are the most important thing for any doing any applying any algorithm of machine learning algorithms, because we fetch anything from these features, because models learns data or learns on the features of the data. So these are the features present in this data set, like mean radius, tumor radius means mean texture of the tumor, mean parameter, mean area. So these are the features in our data set. And cancer data means this column basically give you the actual data which correspond to these features. So these are the data value. Now you want to check the shape of the data. Shape of the data means कि you want to check how many rows and how many columns are present in your data set. So cancer, you have the column data dot shape. So it basically give you the shape of this data column. In the data you have 5 6 9 rows and 30 columns. Now just I this is basically give you in this format, but I want into the table format. You change this one into the data frame format. Df cancer pd dot data frame np dot c cancer. Np dot c basically use np dot c slash np dot r slash. So on which basis you want to convert this data? I want to convert on the column basis, not the row basis. I want to convert into the column basis. That's mean texture, mean parameter, just using c underscore. Cancer name of this one and the data can or I want cancer target, ki target means 0 or 1, or column is equal to np dot append. Np dot append means कि you want to append something. Cancer feature names कि column name is mean radius, you want feature names comma target. So after applying this, the df dot cancer dot head कि I am putting head. The head means कि you want to show the top 5 records. So this give you कि mean radius at 0 1 2 3 4th index. You have 5 rows and 31 columns, in which 30 are the number of features, and last column 1 is the target. Now df dot tail, we already know tail give you the last 5 records. And np dot array basically it is just for explanation ki what np dot c give you, ki if you apply np dot 1 2 3 x dot shape, so is this shape is 3. Now example we take np dot c underscore np dot array 1 2 3 comma np dot array 4 5 6. So example shape is 2. So it basically give you this way: 1 4, then 2 5, and 3 6. So 1, then first one, then is 4, convert into the column, then second, then fifth, that is the second as a second row, then it is come into the column. So it basically conversion into the column type. Now the number of targets in every target, you want to check ki how many of target, ki how many of malignant target and how many of benign target. So you want to check to values. So df cancer target dot value count. So value count, in value count it give you 1.0, that is the malignant. We have 357 data, or we can say that 375 features total data, and 0.0 in the total data 212. Total data we have the 560 something 569. In the 569 we have 357 malignant and 212 is benign. So now total number of targets is 569. You want to check unique number of target. You have only two number that unique, that is 0 and 1, because this is the binary. If you want to check unique number in each column, so mean radius, in the mean radius feature you have 456 unique value. In the texture you have the 479, and the so on. You can check the unique value in your each column. Now, full summary of your data. You want to check the full summary of your data. dfcancer.info give you the full summary of your data. Give which type of your data it has. Mean radius is a float type and not null value. And describe basically give you the stat summary of your data. In the introduction of pandas we already discussed. Describe give you the stat summary of your data, that means count, mean, standard deviation, minimum of all, and max. Total number total null values in your data. At present we load the data. In this data there is no null value, or this is a already pre-processed data. If you want to check ki how to handle the missing data, you can go this website, and in this website different way of is basically discussed ki how to handle the missing values. Now you can check dfcancer.dtypes. You want to check the type of the data, you can use the data frame name dot dtype to check the data type of the data. Unique data in each column. Ki if you want to check in each, so you can also another upper way we apply for the unique data, but now you can check one by one. This give you the unique. In this case it basically give you unique, uh, it basically give you only number, but if you apply iterate for loop on this case, so it basically give you the values also, ki which values are in the column. It is too lengthy. Okay, now if you want to check the duplicate values in your data, you can check the data frame name dot duplicated dot sum कि how many number of duplicate. So I already told you कि we load the data from sklearn. This breast cancer data is already pre-processed. That's why in this data there is no null value, no duplicate value. But when we download from any websites, like we download from the TCGA, so in this case, maybe where we found some missing value, some duplicate data. Suppose we take a sample from same cases but different way. So basically give you the some duplicate data. So you want to check, so you can use this way. Now, if you want to check the correlation among the features, among the columns, so you can use the data frame name dot CORR that used for the correlation. So it basically give you the correlation between the features. So it mean radius, the correlation is higher because features are same. But mean radius and the mean parameter also have high correlation. If we check mean radius and the mean area also give you the high correlation. So that basically if you find the correlation of your features, so you can use this function. Now, if you want to check the covariance between the set of variables, you can use co for the find the covariance between the set of the variables. Okay, now visualize the data. So it for visualization of the data, you all know we use two type of library. First one is the matplotlib and second one is the cbone. Here I am using the cbone, import cbone as SNS. So in this case, I am visualizing the data or each features like mean radius, mean texture, mean area, mean parameter, and mean smoothness. And the hue is the target, that is the orange, basically define you. Okay. Now, orange basically for the 1.0, that means ki malignant, and blue denotes to the benign target. Now, in this case, this pair plot basically visualizes the relationship among the five components, which are the important feature, and compares their pattern for the benign and the malignant cases. Now, the diagonal plots, diagonal plots show how each feature is distributed, while the scatter plot reveals how the pairs of features are correlated. These scatter plots basically denotes at that how pairs of features are correlated with each other. We clearly see that the malignant tumor tend to have larger mean radius, that is the orange, that have the larger mean radius, and the mean radius and the mean area and the mean parameter, and which suggests that features are strong indicate these features are the strong indicators for the cancer classification. Now, overall, this visualization help us to understand the data structure before building a machine learning model. Now, this one basically give you the plot, give you the total count of the target. So this give you the target for the, that is the approximately we check the 2 5 something, or this for the 3 5 9 something, or 2 1 7 something. So it basically give you the count. So you can check by using the um, bar graph. Now you have the another thing, that is the again we have the scatter plot. If you want to plot the scatter plot between on the correlation of the mean smoothness and the mean area, so this scatter plot basically shows the spread of the mean radius of the tumor in the data set, and most values lies in the first and the third, excuse me. This basically give this box plot basically give you help us to understand the distribution of the tumor size and detects some outliers with the disjoint, helps us examine the relation between the multiple tumor feature and how they differ from the benign and the malignant, benign and the malignant. This plot and this plot are the same, but this is the scatter plot, that is the box plot, and this is the LMP plot that is basically use the trend line. Now, if you want to plot the correlation graph, so then basically correlation heat map, then you can use SNS dot heat map df cancer dot cor and a naught is equal to 2, and this basically show you the correlation heat map. Now this basically use for this box plot used to find the outliers in your data set. So basically the this provide you, this box plot provide you the shows that the spread of the mean radius of the tumor in the data set or most of the values lies in the first and the third quartile of the data set and indicate whether the majority of the tumor lies, and the points on the right side basically represent as the outliers and suggesting a few tumor have unusually large radius as compared to the rest. So basically overall this box plot help us to understand the distribution of the tumor size and the data set outliers, while the pair plots help us this pair plot help us to examine the relationship between the multiple tumor features and how they differ from the benign and the malignant cases. So this is about the data manipulation and the data visualization as a huge case study with the breast cancer. So these four steps are the for the area analysis, that is called exploratory data analysis. After doing the exploratory data analysis, we go for the machine learning algorithms. So in this session we just discuss about the data manipulation and the data visualization. The further steps for the machine learning algorithm, splitting the data set, classify the model. Now we switch for the our last from my side, the last topic, hands on the AI biomarker discovery using the RNA binding protein in the cancer. Okay, here I am using the data set from the TCGA. For the TCGA, I show basically show you from where I download the data set TCGA. GDC portal. Okay, for the downloading the data set, you can use two or three way. First one is the you can directly download from the website. Second way is you can use any programming language to download the data set like R, or you can use the Python for download the data set. I am just how I just want to show how you can download the data set from the TCGA. So I'm just showing just simply show you how to download. So for example, I want to download the RNA-seq data just as the hands-on. So I am selecting the RNA-seq data, and the next one is the step is which type of data category. So I am I want transcripting profiling, so I'm selecting this one. Then you want data type, so this is the gene expression quantification. I'm selecting gene expression quantification. The format is TSV, and select for the workflow, that is the star count. Now check for the open access or the control. For the control, you basically give the permission from the website, but I'm taking open, that is checklist for the open. Now which type of tissue type or the other things you can also select as per your choice. For this, now again uh, check for the which type of data you want. So primary sites is, suppose I took the breast cancer data, to select the breast. Now check for the project. I am taking TCGA BRAC. Tick for the TCGA BRAC, and then program is TCGA and less. Now you have seen the TCGA BRAC transcripting RNA-seq data, and format is TSV. This is the sample case 1, 2, 3, 4 and on. And you have total 1231 files. For, means you have 1231 cases which are open. You can download all, or you can download 10 cases, 2 cases for the learning purpose. So in hands-on, I am taking only 10 cases for the learning purpose, because in the our system I have this sufficient memory. So basically, if I want to work on the large amount data, we basically work on the server or the GPUs. So this is the idea ki how to download the data from the TCGA. Okay, or for the RBP, RNA binding protein, you can use IMRG database, from which in the RMG database you have the data RBP list or expression matrix for 13 cancer types. Okay. This is the original website of the IIMRG. Okay, in the IIMRG, you have different type of data. You basically, if in the hands-on, I am using RBP, the RNA binding. So down, you can go downloads. So in the downloads, you have the enrichment of regulation with these related pathways, and you have the correlation between regulation and different immune cells, and you have the expression prediction of regulation in 17 cancers. So which type of RBP you want, you can download from the website. So I am taking from this data set, from this data site, and map with the TCGA BRAC data set. Okay. So basically, I also provide you the sample which I used in this hands-on session, which are already shared by the Amresh sir. So basically, I am using this one, this data set, 10, I'm using 10 cases of the RNA-seq data, and using the TPM unstranded based. So to basically read the files, I'm just using import pandas, import numpy, two libraries for the data manipulation, import globe. Globe is basically globe and the OS, import OS. These two libraries are used to work your system, to basically access your directory in your system. So I am using globe library to access the directories in my system. So files is equal to globe, and this is the path where my tsv file, which is I converted into the excel file, is present in this folder. So basically in the first step I read the files. So in the files I have 10 samples total. So basically give you the details of the samples, key which type sample 1 2 excel file. Now I want to store all the files as a list item with the gene name and tpm unstandard, because I want the to combine whole data in a one file. So for all cases, so basically I read the file and read the excel this f, and this one split by the this one and replace the excel, and in this I have on to add the column name is gene name and the column name is tpm unstandard, and set the index as the gene name. Ki gene name is as a index and the columns is equal to sample. Now append the data into the list. Now length of the list is 10, and if I check for the 5, then xpr list 5, it gives you basically ki gene name and their corresponding samples. Okay, now I'm merging this rows and the column into one. So pd dot concatenate, pd dot concatenate basically used to merge the objects of the data frame. So I want to merge the object, the expression list objects, or axis is equal to 1, that is the column base. Now after the merging, I have this data. This is the name of the genes, this is the row, and this is the samples of the data. So total we have the 10 samples. Now this is the original file. The shape is we have 60660 is the number of rows and 10 are the number of samples. Now visualize the data distribution. Distribution of row tpm value is just right squared and the straight line. That means ki at present this data is highly right squared data. So basically if we have the highly squared data, just we have to apply the normalization, we have to transform the data. Now for the transformation we apply the np dot log to TCG all plus 1. So now we apply the transformation on the TPM data, and now the shape is same, but now data is changed. The data value is changed after the transformation. Now visualize the distribution after the normalization, that basically log transformation. Now this data is normalized. And to check the outliers, we basically plot the box plot of the data set. In the box plot, this give you the samples in which if outliers is present or the not. So basically in this case we apply the box plot, but we can say that on the stat basis we have the outliers. These are showing as a outliers, but in the biologically interpretations here there is no outliers, because the RNA seq data which all are in a sequenced manner, all have a proper distribution of the samples. So again we show the heat map of top 200 variables. So that basically give you the top 200 most variable genes. So it shows that the heterogeneity of the tumor in the data set. It basically denotes the gene name, it is the sample. So this color basically denotes the heterogeneity of the tumor in the data set. Now, after loading or visualizing the data from the TCGA BRAC, we load the RBP file, which is downloaded from the IMMREG database. So in this case, the RBP differential expression matrix are in the text format. You can adjust your path according to your system path. So in my case, this is the path of my file. Now read this file. This /t denote as the tab. If you have the CSV file, so separation is comma, and if you have the text file, that means separation is the tab. So basically pd.read CSV, read the RBP file, separation is tab because we have the text file. Now we want to check rbp.head. So we have the this type of data in the RBP difference expression, in which we have the different type of cancer and RBP ID, RBP symbol, p value, p adjusted value, log FC value, and the test type. So this basically give you the RBP expression matrix. Now filter only for the BRAC related RBPs, means ki in this case we have the 17 cancer, so and we mapping for the only for the BRAC, because our RNA-seq data is the related to the breast cancer. So we basically filter the RBP on the basis of the BRAC data set. So in the BRAC data set, the RBP cancer is equal to BRAC. In this case, ki RBP data frame, that is the cancer in type, because we have the cancer column. In the cancer column, if is equal to BRAC, then total BRAC RBP, what is the length of this one, and print the RBP underscore BRAC, this variable name, dot head, that is the data frame dot head. The total BRAC RBPs are 1435, and this give you the expression matrix of the related to BRAC cancer and on the all these things. Now we get only the gene list from the RBP symbol. So we check for the RBP symbol dot unique and RBP genes and top 200. So in the BRAC RBP, we have these type of gene symbol which are present for the BRAC cancer. Or then we have filter our TCGA matrix to this only to the BRAC RBPs. So that simply BRAC RBP, we basically apply the intersection between this RBP BRAC gene symbol and the TCGA gene symbol. So we apply the intersection between the both because we want to find the common from the both. Then filter the matrix shape, that is the final shape is 1432 comma 10. We have the 1432 total filtering common, which are these are in the RBP and these are also present in the TCGA. At 10 are the number of cases. Now this is the heat map for the RBP heat map. This is the TCGA BRAC sample and these are the sample with the RBP's gene symbols. So now if you want next step, this is for the data. Now you want to find out AI based biomarker discovery, specifically ML based. So first thing is we put the dimensity reduction. We have in the medical data, we have the high dimensional data. So we want to put we want to apply the dimensity reduction. So we just apply the PCA. In this hands-on, we have the small data set, but when we want to work with the real time problem, we have large amount of data. So in this case, I already told you कि in the sklearn, this basically sklearn library is important for the machine learning. It give you the dot decomposition import PCA. So you can import PCA from the sklearn. You can also import the standard scaler from the sklearn for the pre-processing. Now in the TCJ we have the data as a genes as a row and the samples as a column, but samples are very few, only 10 samples. So why we apply the PCA on only 10 samples? We want to apply PCA on the genes to reduce 60,1432 genes, to reduce the genes on the basis of PCA. So just transform to make it as a patient as a row into genes as a column. Now apply the pre-processing, that is the standard scalar dot fit transform to apply, then pass the X, which is the transposed matrix. PCA, that is the n components is equal to 2. So PCA dot fit transform, that is the X scaled to basically apply for the finding the PCA. Now after applying this PCA and after applying finding the explained variance ratio, we want to find out the scary plot. This scary plot denotes that in our data set almost all the data which have highly varianced in the first PC1 and the rest are the linear. So we can say that ki PC1 here in this case PC1 is the more important and rest are the less important as compared to PC1. Now this is the cumulative variance plot. This is the PCA visualization in which clearly see that in the PC1 we have the 9, in the PC2 we have only 1. So for clustering, so 9 are the same group and 1 are in the single group. So here one point is to be noted कि 9 samples in the same group and 1 samples are in the another group, but it is not feasible due to imbalancing. कि if you want to apply the other techniques, other algorithms or further processing, it is impossible, it is not feasible because the groups are imbalancing. So there may be this for one reason we have less number of samples, we have only 10 samples in our data set. If we apply these things on the large amount of data set, it properly works. I already applied on the TCGA GBM data set, differentially gene expression data, normal versus primary or recurrent versus primary, and PCA and K means effectively work because on this data set I have 175 samples and 30,000 something genes. So basically I have the large amount of data, but in this case I am just showing how to work with the machine learning algorithms. We just taking the few samples for the learning purpose. So here there is a imbalancing. So for check for just be manually grouping the data for further processing. I know this is not the statistically right, but for learning purpose ki how to apply the other things after grouping the patients into the different on the basis of their characteristics. So I check for the PC1 and the PC2. So check for the PC1 and PC2 and sort the value. The data are in this format. So now we check that all the data, most of the data are present in the PC1. That means ki this is the PC1 and only this one is not in PC1. So we find the median based on the groups and divide the data on that basis. So we group the data on the A has 5, B has 5. After finding the group, we find key RBP biomarkers on the basis of the AI feature for the AI feature discovery. For this, identify which RBP distinguishes the two groups. For this, we apply for group A and group B. In group A, we have this type, and group B, we have number of genes and number of sample is 5. After that, we apply Mann Whitney U test for the robust for small n. If we have the number of sample is small, then we can apply for this. You can also try T test for the differential gene expression, finding the differential gene expression. After applying this, we find the p value or log fc. And after that, we also applied multiple tests for FDR correction. Multiple tests for FDR correction means FDR is used for false discovery rate. But in this case, I'm applying this one FDR and find gene, log FC, p-value, and FDR. Now the key observation, all genes have FDR, that is the 0.568, and it is due to very small sample size, that is the n is equal to 10% and large number of gene tested, and the strong multiple testing penalty. So that means ki, there is a lack because we have a small data set. Now after applying this, to find the top significant genes, we take p-value which is less than 0.05 were selected for the further downstream stream analysis. So in the DG result, if we have, so we have this type of data. So significant genes in the 75, we found 75 genes are the more significant in as compared to 1432 genes. So we find the 75 as a significant genes, and we also plot the volcano plot for the p-value, and after this finding the significant genes, we link these genes to the mutation data. This mutation data is also downloaded from the TCGA BRAC, that I have already told you ki from TCGA BRAC, just selecting instead of RNA-seq, we select for the mutation data. After how then combine also for the mutation, I also take the same mutation data for 10 samples, and I have this type of data, then check for the top one, then compute the tumor mutation burden, that is TMB, and check for this one, that is the tumor sample barcode. And after applying this, I am checking for the TMB index and cluster DF. So after checking with the index and this one, I am mapping the sample names and the bar codes. So case to barcode, this is the TC BRAC sample 1 and this correspond to this one. So I am mapping this one as so on sample 10 we have this sample barcode, sample 2 we have this sample barcode. Now compare the TMB between the groups, the group A and group B. So after finding the TMB compare between the group, so TMB is 49.0, group B is equal to 64.2. So in one group has higher TMB, support the biological relevance and clinical relevance of the group B. After this, visualize this TMB difference. In the group A, we have this one, and group 2B, we have 62. Now, after this RBP2 mutation for the next generalization step, we check for the Hugo symbol from the mutation file is in the significant gene, yeah, is present to count the value of that genes. So at present, only one gene is which is related to the Hugo symbol from the significant genes. So we can say that this is the one which is more important RBP biomarker. Next step, AI discovered therapeutic candidate genes. We check for the RBP biomarker. We take the complete significant gene list and then map to the KGG cancer pathway to find the functional validation of the significant RBPs using the pathway enrichment analysis KEGG. After this one, we find that we have this type of we have the genes, these are the associated with the cancer pathways, and these are the gene set name, that is the splisho and the RNA transport, which are further used for the personalized medicine. Plotting the top pathways, the ribosome and univariate mediated RNA transport, RNA degradation. So this is for a learning purpose how we apply the machine learning algorithm PCA K-means to find the significant RBP biomarkers from the RNA binding protein data. For personalized medicine, we can further consider that we can check for the patient specific prediction, stratification of patient, personalized risk group, and treatment targeting based on the RBP profile. Now in this hands-on session, I specifically applied the PCA and the K-means. For PCA for the dynasty reduction and K-means for the clustering. But if you want to work with the deep learning approaches, you have the you can also same use RBP expression matrix, but you can use autoencoder instead of PCA for the dynasty reduction. And for group patient, you can apply k-means clustering on the learned latent space. That learned latent space is basically provided by the autoencoder, and find the important genes, the feature importance, and the model interpretations. And that's all from the my side. And the second session for the second use case basically will be discussed by the Priyanka Kataria. And from my side, if you want any question, then you can ask me. If you have any question regarding the pandas, regarding the data manipulation and visualization, regarding to this one, so you can ask me. Yes, if you have any question, then you can ask me. Otherwise, I will hand over to Miss Priyanka Kataria for the next session. Shivani Bhargavi, my mail ID, consider this is mail ID. Okay, Siva Bhargavi, I will ping you on the provided mail ID. Okay, no one have any question? Priyanka, are you there? Priyanka?
प्रियंका को कॉल कर लो एक बार.
Okay, sir.
Yeah, hello Sapna. I'm here. Hello.
हाँ
Okay.
प्रियंका, अब आप कंटिन्यू करो आगे.
Okay, sir. Thank you.
Okay, I'm stop sharing.
Everyone able to see my screen?
Yes, it is visible.
Okay, thank you, sir. So, good afternoon, everyone. I'm Priyanka Kataria. Today, my topic is application of machine learning in drug design targeting RNA binding protein in cancer. So, now I am starting from some core libraries and toolkits. Here are some core libraries which are already learned by Sapna Yadav. And there is one new toolkit, that is RD toolkit. RDKit is a free open source software toolkit used in computational chemistry and chem informatics, where we can work with chemical structure on a computer instead of doing everything by hand. So we used it here. These are some machine learning tools and techniques. So first we install all these, then we start our coding. As a input, first we input file as a smile list of a chemical structures, which are the text representations of molecules. So here data is chemical text, but we know machine learning cannot understand chemistry directly. So we used RD toolkit molecules to convert these these are the smiles, as a object. Like which kind of object? Here we convert these descriptor. These are some descriptors which are extracted by using RD toolkits. They calculate its properties from what we input. Like we input as a smile. If we input, sometime we input small molecule as molecule files or SDF files or smile files. From that, we calculate molecular weight, number of atoms, log p values, hydrogen bond donor, acceptors, etc. So in my coding, we calculate some basic features of these. These are the basic features of these, which are highly recommended for further binding affinity calculation. As we given some smiles as an example, these 5 smile as an example in list, we calculate one by one their log p value, molecular weight, hydrogen bond, acceptor, donor, and polar surface area. So these are the values we calculate by using RD toolkits. After that, we visualize these descriptor by using plot. These are my ligand index and these are the molecular weight. Same type, we distribute our log p values in figure as these are the log p values of that molecular descriptor and these are the ligands. So same kind, we can distribute so many plots from that in Python by using machine learning. Next, we calculate the features of the smiles or as we say descriptor by using machine learning. We scale our features. Why we need scaling? Because all descriptors are not comparable range. So machine learning cannot be scale in very but basically machine learning is very sensitive or critical, which are sensitive for scale. It would bias toward large number of data. Like here, molecular weight are very high as compared to other values. So that's why we need scaling. If we don't do scaling, our machine learning performed very poor. So we transform our data and scale our data. Then we binding the affinity labels. These are some examples of binding affinity labels. Are not actual. We use it as a demo. But here we can use actual data. After that, we train our model. Here we used random forest model. It learn relationship between molecular descriptor and binding affinity. Here, why we choose random forest only? Because it is a non-linear model and it is robust, widely used in chem informatics. So we can use another model also, but it best fit to our above example. After that, we find the predict by binding affinity. Here are the actual values of our binding, and these are the predicted values. As you've seen, there are very familiar values between our actual and prediction. So, what machine learning do is here, when we give our actual values, after some time, when we need to next value, then machine learning predict the next value on the basis of these actual values. See, these are my predictions and actuals, which are most commonly nearby the actual lines. These are the very basic or best predictors. So to make this work in real research work, we get the binding data by using binding tools like binding DB, Campbell, and we can use thousands of ligands. We can add protein encoding. Also, we can sequencing our data by using CNN and transformer. Also, we use here some protein structure and contact map like graphical neural network. In our process, we also used graphical neural network in next step. But before that, we use some most important, that is Morgan fingerprint. Why we use it? Because it convert molecules into a fixed length of binary vector and capture the chemical environment which are locally and most widely used in ligand encoding in machine learning and QSAR. So these are the Morgan value calculation of molecules. So machine learning cannot understand chemical structure directly. That's why we use Morgan fingerprints and it converts the molecules descriptor with its fixed length of numerical vectors that are represented in chemical substructures. First, we create from the above as an array for the efficiency in machine learning. These are the visualization of fingerprint density. This plot shows how well this model fits in it. This is not a real validation and not a real fingerprint bit matrix, but as an example, we uh, we show how our ligand and fingerprints are shows here. See, rows are ligands and columns are chemical features, and these bright spots are active substructures. This helps us to identify shared motifs, unique chemical patterns, etc. After that, we apply PCA, that is a type of clustering. Why we use it? Because it reduces our 1024D data into 2D data. But above fingerprint do, they give us 1024 dimensional data. And what PCA do? It convert the above 1024 data into 2D forms and project molecules into a latent chemical space, and also interpret nearby point into as a chemical similarity and distant point as a chemical difference. मतलब it means chemical are far or dissimilar to that chemical or that structure. It is a basic part of the clustering or diversity analysis. See, here are the some similar chemicals. These two are most common, matlab, it means most similar kinds of chemical, and these are dissimilar from these two. Next, we do graph neural network. What graph neural network do? It convert molecules into a graph. Each molecule become a node as an atom and as is as a bond. So what it do? It present our molecule as atoms are all nodes and which are connected with as bonds. So here atom get its numerical values, its total degree, its formal charge, and aromatic values. These are the atom features which are written as a atom molecular features. Here why we do it? Because it gives us naturality of graph, because as we know molecules are naturally not graph, they are present as a vector here. Next, we convert molecular graph conversion. Here what we do, we convert GCN convention data mass is passing and global man pool which are graph embedded, and then linear which are affinity predictions. In this step, we did atom to atom interaction, structural contact, and non-local chemical effect. This is deep learning based ligand encoding. Then we did each ligand in a graph network. This is the simple model which is follow the above rule. Here we pass feed forward network, which is deep learning part. This one is deep learning part, feed forward neural network. Then we forward pass. This is a demo prediction, not an actual. That's why we skip the some steps here. After passing the forward neural network, we find out the predictions affinity. Here, forward pass shows the graphical neural network can output as a prediction affinity demonstrate end-to-end learning from the structure. So in this work, ligand were encoding using both traditional fingerprint-based representation, and graph-based neural network encoding. Morgan fingerprint capture only substructure patterns, while graph neural network learn atom-level interaction through message passing. These learned embedding were used for binding affinity prediction, forming a scale form framework that can be extended to protein ligand fusion models. So, in this program, we start from the smile, then we extract the feature of that smile, scale that feature, then apply machine learning, and then predictions. From that predictions, we use some more values like Morgan fingerprint capture substructure patterns. Then we also use deep learning structure as GNN and feed forward neural network for message passing. Overall work, what it do? It done virtual screening. It helps in binding affinity prediction. It also used in protein ligand modeling. If we did this all the if we do all this work in a proper way, then we can use it as a kind of work like molecular descriptor, Morgan fingerprint, and state of the deep learning in our drug designing. So have you any question about that? Actually, this is a demo. If we use the actual data here, we struck because we need GPUs or processor for it. So we didn't use here the actual list of smiles. We used only 5 sample. We don't use any fingerprint Morgan values here. We used only examples. We don't use here any KD or affinity actual values. We used only 5 samples. For that, if we do it in proper way, we definitely find some proper values of these patterns. What are the affinity, which ligand are best for binding, all these. Thank you. Have you any questions?
Thank you, Priyanka and Swapna. I have one question. Like you are using a Google Colab and Sapna is using a Jupyter notebook. So suppose if we do not have any coding background, and if you want to use them, can we use directly? And how can we start? Like what is the basic beginner level of learning, like machine learning?
Ma'am, can I answer this question?
No.
Yes, ma'am. Can I answer this question?
Yes, please.
Okay. Okay. These are the IDs for doing the Python related task like Jupyter Notebook and the Google Colab. The Jupyter Notebook is basically used for the local systems, and Google Colab is used for the clouds, on the cloud. One basic difference between the Jupyter and the Google Colab is कि if you are working Jupyter Notebook in the local system, it basically works on the CPU. If you using the Google Colab, it basically also provide some facilities like GPU and the TPU. If you are using the free version of the Google Colab, it give you for the some time like 2 hours in one day. You can utilize the GPU and the TPU facilities. So it basically depends on you कि which type of work you want to do. If you need GPU and the TPU, you can use Google Colab. The syntax and way of writing the code is same in the Jupyter Notebook and in the Google Colab. If you don't want to use any ID like Jupyter Notebook or any Google Colab, so you just simply install Python in your system. After installing the Python, I will show you how to install work with the simple Python. So basically, okay. I will show you कि if CMD. Okay, you don't want to use Jupyter Notebook, you don't want to use the Google Colab, and also you don't want to use the any ID like the Visual Studio. So in case you just download the Python. I have already installed Python. Then in case I'm using Python, this is the Python interpreter. There is no need of Jupyter, there is no need of Google Colab. Just use the Python, and this is the Python interactive mode. In the Python interpreter, you can write your code. For example, I am writing a is equal to 10 and b is equal to 20. Just writing the code as per your syntax, as per your work, like c is equal to a plus b, and you want to print c. So just enter and it give you the result. This is the one way of using Python. You can write the code here without using any ID. If you want to write the multiple codes and you also want to save this file, so just use any text editor like Notepad, Notepad++, or any other text editor. For example, I am just writing using Notepad++. Here you can write your whole script. I'm just writing like same code. A is equal to 10, b is equal to 20, c is equal to a plus b, and then print c. So it basically, and save this file. This file, suppose I'm saving in the documents trial, and the for the Python you just you can use the python.py extension and trial. So this file is basically saved in my local system, that is in the documents, that is the trial. Now I want to execute this script because scripting mode is better as than to this interactive mode, because in the scripting mode you have you can write the thousands of line of code and save the file. After 1 month you want to use this code, you can just use this code by just I'm just exiting from the Python interpreter. Here write Python, then the path of this file. I'm just copy as path, and then Control V paste, and it give you the result. So here I have just one output. If you have thousands of code and according to you apply the print, you apply the plot method, so it basically give you accordingly the results. So just this is the way you can start from using the Python. But I suggest you if you want to use Python as